afternoon, ladies and gentlemen. First of all, I would like to thank the scientific and the organizing committees for select our work for oral presentation. As the title suggests, my presentation is about the evaluation of the global molecular surveillance of the human respiratory syncytial virus in the context of the vaccine implementation. Well, since the first work in the matter in the 90s, the molecular epidemiology of RSV has been developed so much until now. 13 genotypes have been described for the subtype A and 20 genotypes for the subtype B, and depending on the author and the reviewed paper, the criteria used for the genotype definition varies. On the basis of phylogenetic analysis, some authors use the ectodomain region of the G gene, others use the second hypervariable region of the G gene, even other genes like the F-glycoprotein gene or the 3-surface glycoprotein genes. And recently, some authors use for the classification of the strains the complete genome sequences. And in addition, a duplication of a segment in a G gene has been used for the genotype definition. And some authors use the evolutionary distances, also known as genetic distances, for support their definition. And the first question is, depending on the portion of the genome used, is the classification of the strains equivalent? Are these analyses comparable? And taking into account the great progress on the vaccine development, it's important to compare data among different countries before and after the vaccine implementation. For this reason, we wonder, can we unify criteria for the genotype definition? In this work, we determine the best region for the genotyping process, and we propose a cutoff value to define genotypes on the basis of genetic distances. And finally, we propose an algorithm for global molecular surveillance at different levels. How we did proceed? We download all the complete genome sequences published in GenBank to date, and after applying some filters like the elimination of the duplicated sequences, we obtained 2 alignments for both subtypes. As we could obtain complete genome sequences in our laboratory by next generation sequencing directly from random for clinical samples, and random amplification, we include those sequences on the analysis. And well, we perform phylogenetic analysis by different inference methods with different portions of the genome. We use the complete genome sequences, the 3-surface glycoprotein genes, the G and F gene, the F gene, and the ectodomain region and second hypervariable region of the G gene. In this slide, you can see as example the phylogenetic trees obtained for the subtype B by Bayesian methodology with different portions of the genome, of the complete genome. And as you can see, the topology of the phylogenies are similar, but we can determine that the best resolution for all the clades with high statistical support was obtained with the complete genome sequence. But beside this technique still being quite expensive, and currently exists low representation of the circulating strains, not only in the number of the sequences published, in comparison with the G ectodomain region sequences, for example, but also in the countries of origin, as you can see on the pie charts. And for this reason, we try to find the minimum region to genotype the strains. And as you can see on the loops, the statistical support remains high in all the trees, with exception of the last one, which corresponds to the second hypervariable region of the G gene. This led us to think that the minimum region that can be considered for the genotyping process should be the G ectodomain region. Well, but At that point, we have to deal with other important problem. While some genotypes always show well-defined in the phylogenetic trees, other genotypes do not, and it's difficult to assign a classification for new strains. We tried to solve that problem, and we found a work published by Trento and colleagues last year in which they analyzed the variability of the G-ectodomain region of the subtype A strains. And on the basis of genetic distances, they define a cutoff value. They use the genotype GA1, the genetic distance within GA1 genotype as a cutoff value for the definition of genotypes. But they use they genotype because this is the oldest genotype and which has no longer been detected in the population. However, some old strains have been published after their work, and for this reason we update that analysis. We download all the G-ectodomain sequences published in GenBank. We obtain 2 alignments for both subtypes, and for subtype A, 43 sequences classify into GA1 genotype, and the genetic distance within this genotype was 0.038. In the case of subtype B, 21 genotypes classify into GB1 genotype, and the genetic distance within this genotype was 0.038. Well, therefore, at the bottom of the slide, you can see our proposal of genotype definition with the G-ectodomain region, a well-defined phylogenetic clade with high statistical support and an average genetic distance below 0.038. And taking into account our proposal of genotype definition, we reevaluate our phylogenetic analysis. We estimate the evolutionary distances between and within the genotypes previously described. And we found some discrepancies. In the case of subtype B, NA1 and NA2 genotypes did not show us a monophyletic clades. And in the case of ON1 and GA2 genotypes, the genetic distance between these genotypes was below the cutoff value. For this reason, we propose that ON1 genotype and NA genotypes could be lineages within GA2 genotype. And as a consequence, this should be the final classification for the subtype A, as was described by Trento. And in the case of subtype B, recently reported sequences dated from the 80s fulfill our genotype definition. And for this reason, we named as a new genotype GB5. And in addition, the genetic distances among BA1 to BA10 genotypes were below the cutoff value. And that's why we propose that BA1 to BA10 could be lineages within BA genotype. And similar results were obtained with SAB2, 4, and GB3 genotype, and with GB2 and GB4 genotype. And as a consequence, this is the classification for the subtype B. In addition, other interesting result was the behavior of the topologies of the phylogenies of the 3 surface glycoprotein genes independently, in comparison with the complete genome. The topology of the G gene and the F gene shows similar to the complete genome. This is as example. This is the phylogenetic trees obtained, for example, for subtype A by these some methods. It's similar in the case of subtype B. And in the case of SH gene topology, the topology of the tree is completely different. And taking into account that the G and F proteins are the main antigens triggering neutralizing antibody response, we wonder if the selective pressures on them are similar. And by bioinformatic tools, we analyze the selective pressures acting on them, and we found that, we found for the both all the time, we found that for both subtypes, the F protein only present negative selective sites, and G protein present positive and negative selective sites. And that's why we consider that both genes should be monitored in the future, not only for the classification purpose, but also for their behavior after the vaccine implementation. And finally, I would like to summarize all the work presented here, showing our proposal of algorithm for molecular surveillance of the virus. After each annual outbreak of RSV, 25% of the samples should be selected at random, and for each of them, the nucleotide sequence of the G ectodomain region should be obtained in order to genotype the strains, taking into account our proposal of genotype definition. And taking into account too that the most advanced vaccine is based on the live attenuated strain, it's important to monitor the evolutionary process on the whole genome. And for this reason, we propose that a portion representative of the detected genotypes should be selected for the complete genome sequencing. Well, this brings me to the end of my presentation. And I would like to remark that a unified criterion for genotyping will allow us to compare the circulation patterns globally and locally before and after the vaccine implementation. And all this information together will help us to know how the vaccine implementation could affect the ecology of the RSV in the future. Finally, I would like to show you all the virology laboratory team of the Ricardo Gutierrez Children's Hospital in Buenos Aires. Thank you for your attention.
Thank you very much. Very nice presentation.
Thank you.
Are you able to designate some of the sequences from the reference strain in the genotypes so that everybody can use the same reference sequence?
Do you say that some sequences always show as a reference for the genotypes?
Yeah. So if some of the sequences within each genotype are labeled as the reference strains or reference sequences for that genotype,
yes. I think that with our suggested genotype definition, it doesn't matter if one sequence is a reference or not, because the definition supports that a group of sequences, as you consider a clade, in the analysis of the genetic distance, these analyses support your suggested genotype or not. In this case, I remains the name of the genotypes, but the important if is these sequences are a genotype or not, with the genetic distance support.
Well, very brief. First of all, I want to thank you because my name is Teresa Perret, and me and Larry Anderson established the genotyping GA1 through GA7 and GB1 through GB4. So you took me back to memory lane. Where does the South Africa genotype, the one established by Venter, where do you think that would fall? Would you, under your criteria, that would fall as a genotype or as like a stray from what's established?
Can you repeat that?
No, would you consider the South African, the one that has been only described by Venter, as a genotype or like some outlier back on the sampling that she had at the time?
Yes, I consider all the genotype describes, the historically genotype describes. I consider the genotype described by Venter, but she used the second hypervariable region. Mm-hmm. We consider in the work the second hypervariable region, and the support decreased so much. For this reason, in the analysis, I can include the sequences of Venter when consider only the second hypervariable region, but I consider that the NA genotypes should describe the NA1 to NA4 genotypes. And I consider that maybe these genotypes could be lineages because some strains classify, when I consider the second hypervariable region, I don't know if you understand me, but when I consider the second hypervariable region, I include as reference the sequences of Venter. Some strains classify on these genotypes, but when I analyze the Xi ectodomain region, these strains, these genotypes is not a support as genotypes. For this reason, we propose that could be lineages into XIA2, the old XIA2 genotype.
Okay, thank you.
Oh, thank you.